Hello everyone. In this video we are going to discuss about a process which occurs in the cells and which helps the cell to multiply itself. And that process, as you can see, I've written it down right here, is called mitosis. Now, some of you might already know what mitosis is, and for some of you might be new, so if you know this already, that's good. That it will help you guys to review some of the facts which you might have forgotten. If you don't know this, then it's a good thing to understand it for the first time. So let's begin. Now, a cell basically there are two kinds of division which occurs within the cells, by which cells are able to reproduce, or by which cells are able to divide themselves into many cells. And one process is called the mitosis and the other one is called the meiosis. But in this process in this video we're going to focus on mitosis here. Now, before I go into the mechanics of mitosis, first let's just before even the mitosis happens, there is a process through which the cell goes, or the phases which the cell goes in order to prepare itself to enter into mitosis, and that phase is called the interphase. The interphase. Now, basically, the interphase has been divided up into three main stages, or three main phases. So it has been divided up into a G1 phase, then the second is called the S phase, and the third is called the G2 phase. Now, these three phases, and if you notice here on the G1 phase, what basically is I think the G stands for is called the first gap. So 1 for the first and G for the gap. So the first gap. And what it is, it's just simply the stage at which the cell lives its life, or it's the phase during which cell is doing its day-to-day activities, making proteins, living its life, like having a good time. Now, the second phase, which we call the S phase, which means in this phase actually the DNA starts to do the things which he thinks, okay, now I should get started in order to get ready to enter into mitosis. And what happens in this phase is that if you see the structure of the cell, that if this is the nucleus and this is the this is the cell membrane, so within the nucleus you have this genetic material lying around. So I'm going to try to draw only two different kinds of two different kinds of thread-like structures here. So one of them can be, let me give a brighter color. Let me pick this one. And the other one, so let's say this is the other strand. And so I only draw two strands here, but in reality there are about like hundreds of strands. They're coiled up in each other, so you can't really differentiate them. And this length, this strand, or this thread-like structure, you may say, of genetic material is called chromatin, and we discussed this in the previous videos. So chromatin, and just for a quick review, it's always good to review, is that chromatin is basically a DNA, a big chain, a big thread-like structure of molecule of DNA combined with proteins, which are called, well, if you said it by yourself, you're absolutely right, which are called histones. And the combination of these two, we call them chromatins. And DNA, and the histones gives the DNA, this molecule, its structure. They attach to it to give the DNA. They are basically called structural proteins, these histones. So in this part, in the G1 phase, the cell is just growing and living its day-to-day life, and as it reaches to the S phase, the DNA starts to replicate. So replication occurs. So if there's this strand is in this strand right here replicates, and there's another strand right here, and if I were to draw the other one, so here is the other so this is the other DNA strand. So in the S phase, the DNA replicates itself, so it's now preparing itself to enter into into mitosis. So G1 is just a growing phase. When it's the first stage of interphase in which the cell grows. The S phase is where the DNA replicates itself. Now the second stage is we call it the G2 phase, or the second gap, and this is also the cell's growing phase. Cell living phase, in which it's the last stage before cell enters into the mitosis. Now the interphase, and I should have mentioned this earlier, the interphase plus the mitotic phase. The mitotic phase are together, these two combinedly are called, the cell cycle. These two combinedly are called the cell cycle. And the cells, what is this? So you might be thinking, okay, what is the definition of a cell cycle? Well, the cell cycle is basically, it's a cycle in which the cell a series of events occur within the cell through which it grows and reproduces. So this whole process of like growing, living, and then reproducing, dividing itself into half, and then those two halves do the same thing. So the cell cycle is basically a series of events which occur within the cells, and it consists of the interphase and the mitotic phase. And this interphase is divided into three subphases. The mitotic phase is mainly divided into, well, mitosis. The mitotic, so it tells you 50% of it. The mitotic phase, and plus the cytokinesis. Cytokinesis. So as before, I was just reading, before I would make this video, I was just reading this mitosis process, and to be honest with you guys, since from my freshman year and before, in my freshman year when I first learned this process, I always thought when the cell divides itself into two cells, we usually call it mitosis. Like if this is a cell and it divides itself into two cells, this is what we call mitosis. And actually I wasn't right. This is not what mitosis is. And what mitosis is actually, if the nucleus of the cell, like, so let's suppose this is the cell, and you have and in the nucleus of the cell, if this nucleus is divided up into two different nucleuses within the same cell. So if you have this is the nucleus and this is the other nucleus. Now this cell has experienced mitosis, which means that mitosis has occurred. So the mitosis, which the mitotic phase contains one of these parts, is just the division of the two nucleuses, whereas the cytokinesis means the division of the cytoplasm of the cell, which happens after the mitosis. Well, it happens along the same process, but the people who observed it divided into different kinds of ways. Like one is called mitosis. The mitosis only means that the nucleus has been divided. So this cell has just experienced mitosis, and if cells divides up, if this cytoplasm divides up into two cells, and you have a cell membrane right here. So now when you have two different kinds of cells, then we call it cytokinesis. So just for this, it actually, I didn't know this till a few days ago, so it's a good thing to know. Now we were in the S phase, in which the chromatin has divided up itself into two different kinds of, the chromatin in the nucleus has been replicated, and then it goes into G2 phase, which is just a regular growing phase, or the second gap, and now it goes into the mitotic phase, which contains the mitosis and cytokinesis. Now after entering into the mitosis, the first phase is called the prophase. Now prophase is and in prophase, what actually happens is that the chromatin actually becomes condensed and it forms this kind of structure. Uh, yes, it becomes these kinds of structure. Oh, I should pick the same color which I did. The different colors can be confusing. So I just drew, like, if you have two strands of chromatin right here, this one replicated and this one replicated too. So now you have, now these chromatin become condensed. They become like they become coiled up together and they form this kind of structure. So this pink one becomes like this. So this is a chromosome, and the yellow one becomes like this. So now you see this, if this is the original one which is present in the cell, this is the replicated one, and same with this. So now this is the nucleus of the cell. So I'll try to focus on the nucleus right here, not the entire cell. So when I draw the circle, it's representing the nucleus. Now outside the nucleus there are these two organelles which are cylindrical shape, and mine hardly looks like a cylindrical. No, yes, it's close. Now they are perpendicular to each other, and in the introduction to the cell video we discussed about these. This is called a centrosome, and each centrosome contains a centriole which are responsible for which release microtubules which help in the cell in the division, which we are going to discuss. So what in the prophase, the chromatin becomes condensed and becomes chromosome. So this whole structure is called a chromosome, and each of this hand which it has, like this part right here and this part right here, are called chromatids. So this whole thing is called chromosome, and each hand is called a chromatid, and they are attached to each other at this central point right here, both of them, which we call the centromere. So the centromere. Now, so as I was saying, we also have these centrosomes out there too. So now what happens is that in the prophase, the chromatin converts into the chromosomes. Now we enter into another phase called the prometaphase. And what happens is that in prometaphase, this nuclear envelope right here starts to disappear. So I should draw it a little bit down. So in prometaphase, this nuclear envelope starts to disappear. And the centrioles move in the opposite direction. So the centriole moves here, and the centriole moves here, and so the centrioles move here to the opposite direction, and they're trying to move in the opposite direction. And the early spindle formation, the formation of spindle the microtubule spindles, which means like it's like a net, they start forming it around it's just an early spindle formation which is formed around the cell in order to help mitosis occur. So nuclear envelope starts to disappear, and the chromosomes are still present here. So this is one chromosome, and this is the other chromosome. So now you have two kinds of chromosomes, the nuclear envelope, this nuclear envelope starting to disappear, the centrioles are moving in the opposite direction, and the spindle formation is beginning to happen. Then you enter into the phase called the metaphase. Now the metaphase In metaphase, what happens is that prophase, pro-metaphase, then prophase, metaphase. Yes, you enter into the metaphase. Now what happens is that in metaphase, the microtubules attach themselves to the chromosomes. They attach themselves to the chromosomes. How do I draw it? Yeah, yes. So they attach their. So if you have these spindle fibers, so these attaches themselves. They align here. Oh, so this is one chromosome now, and this is the other chromosome. So the chromosomes are attached to this microtubule, and I'm doing it very messy here. So they attach themselves to the microtubule, and if you note, and what happens is that in the centromere you have a molecule called the kinetochore. In the centromere you have a place called the it's a molecule called the kinetochore, where the microtubules attach themselves into it. And what happens is that in metaphase, it's just a process in which the chromosomes align themselves to each other. Now, so, and remember, we still have 2 chromosomes in the cell. So if I were to draw the whole the cell membrane, we still have 2 cells. And now what happens in the next part? What happens in the next part, which we call, so let me draw it here. In the prophase, prometaphase, metaphase, then metaphase, and anaphase. Now, then after the metaphase, we enter into. After the metaphase, we enter into anaphase. And what happens in an anaphase? It's basically the pulling apart of the chromosomes. They pull apart, so each chromatid gets pulled apart. And remember, they are only called chromatid. And each of these hands are only called chromatid until they are attached with this, and until they are in this form. When they are detached from each other, each individual hand is called a chromosome again. So let me just take a little bit of time of you guys to explain this. So you see that if this is the nuclear material right here, if this is the nuclear material all by itself, it is called, in its condensed form, this is called a chromosome. When it replicates and forms this structure, it is still called a one chromosome. And while it is in this shape, it is called a chromosome, but each individual hand, this part right here, this part right here is called a chromatid. Chromatid. And when it detaches itself, so you see like this part gets separated and this part gets separated, now again we call them a chromosome. So I hope I'm not confusing you guys. So this is called a chromosome. This is still called a chromosome with 2 chromatids. When they detach, they're again called a chromosome. So now after the metaphase, it enters into the anaphase. And now in anaphase, now I'm drawing boxes right now. So you still have centrioles on the opposite side, and you have these microtubules. And what happens is that these two get pulled apart. So how does it happen? Well, so you might be thinking like this part right here detaches itself from this one and this one. So let me change the color. So this one right here, this one gets detached, and this one gets detached. Now this one is going in that direction, and the other one is going in that direction. And the yellow one, if I were to draw it on the separate, it would be easier for you guys to understand. So So it look like this. So these two get detached from itself. So now here they were joined together, while here they're detached. So if you notice something, here's something very cool, well, in my point of view, is going on, that you start off with two. Look, if you look at here, this is one, this is two. Now when they are detached from each other, two of them are going in this direction, while these two are going in that direction. So you're still going to end up with two number of chromosomes, well, in this case. So these two get detached from each other, and these microtubules start stretching in on the opposite poles, on the opposite direction, and the cell starts to elongate, or the cell starts to elongate, and the chromosomes are going in the opposite direction. Now, after the anaphase, you enter into the telophase. Now, keep in mind that the cell is still elongating. So, if this was the cell like this, while the process of anaphase, the cell is elongating, and this is the microtubule. This is the centrioles, and here is the chromosome. These two chromosomes are here, and these two chromosomes are, and they're pulling them apart. And so you might be thinking that after keep on elongating, the cell would become something like this, like a pair of glasses that you wear. So you still have these two chromosomes. You have these two chromosomes, and the cells just keep on elongating. Now, in telophase, that's exactly what telophase is. I said it earlier. In telophase, the cell keeps elongating, and the two nucleuses become apart from each other. So this is, well, I just saved it for later to show you guys something. So in the telophase, you see that these nucleus keep on not the nucleus, the cell keeps on elongating, and you have two different kinds of nucleus. The cell keeps on elongating, and these genetic materials are on the opposite direction. So if I were to draw this huge cell which is elongating, so the two chromosomes are here and the other two are here, right? So now what starts to happen is that the nuclear membrane that disappeared in the first part starts to reappear all of a sudden. Starts to reappear, and the nuclear membrane here starts to reappear also. And what happens is during the telophase, you have the nuclear membrane completely restores itself, and the cell has two nucleuses. Now the mitosis is finished. The mitosis, the part of mitosis was to divide the nucleus, to get two nucleuses with equal amount of chromosomes. So here is you might see the yellow one, and here is the pink one, which I didn't draw in pink. That's my bad. So here is the pink one. So this is the pink one, and this is the yellow one. You have two chromosomes on either side. Now, the mitosis is finished now, but the cell which the part which it still needs to experience is called the cytokinesis, and it's the part where the cell is still elongating. And what happens in this part, that as it's elongating, the cell becomes like this. With these two nucleuses, and then eventually they separate. And this part right here, so these two are going, these indentations which you see here are called the cleavage furrow. Now, in the cleavage furrow, so these cells are like contracting within themselves to help itself to detach themselves from each other. So how do they do that? Actually, they have in here, right in the cleavage furrow, you have You have microfilaments made of the actin protein, and you also have myosin. And if you have you might have studied in your biology class that actin and myosin are the same proteins which act into your muscle for contraction. So this is basically the same mechanism. These cells are contracting in this in inward direction, and they're separating at the same time. They're separating themselves from each other. So as they separate, so then you end up with two cells with equal amount of number of chromosomes. So you start off with 2, you end up with 2. So this is the part how basically an animal cells divides. Whereas in a plant cell, you have sac-like structures called vesicles, which are filled with cell plate, and in order to divide the cell. So if this is a plant cell, and you have 2 nucleuses, so cell plate starts forming right here till the cell plate formation is complete, and then it divides the cell up. So in plant cells, you have cell plate starts adding up here in the middle of the cell to divide the cell into half. So this is pretty much it. Now what I wanted to show you guys in the figure is, so I didn't show you the figure first because I wanted to help you try to see that how actually it happens. Now if you look at the figure, this number of figures which I have right here, so this is the centrosomes. These are the centrosomes right here. This is the chromatin which have been replicated, and this is the G2 of the interphase. Now you enter into prophase, where these centrosomes are moving in the opposite direction, and they each are replicating their mitotic spindles, or these are microtubules. And look, these things have condensed, have formed an X shape. Now here they have given you the example of 4 of them. Then what happens in this pro-metaphase right here, you have these spindle formations have completely occurred and have completely been formed, and these nuclear membranes right here are beginning to disappear. You have only fragments of nuclear envelopes or membranes. Now this is called a metaphase plate, which is just an imaginary place right in the center between the two opposite plates. Now look now here is the most interesting part, which I believe which differentiates, where some things differentiates from mitosis are different in mitosis and meiosis. And one of them is this: in the anaphase, when the chromosomes are being pulled into opposite directions, they eventually cut them in half. That's why you have like 4 here, like 1, 2, 3, 4, and they split, and when you're forming 2 new cells, you still have 4. So the number of chromosomes doesn't change at all. So this is one of the key features which differentiates from mitosis and meiosis. So in this part right here, if you understand this, you'll quickly get it, that why in meiosis you have reduced number of chromosomes, and why in mitosis you have same number of chromosomes. So they start pulling each other apart, and in this phase you see that this is part like these indentations right here. You see this? So these are called the cleavage furrow. I wasn't able to draw them as neatly as possible, but here you guys have the what I want to say, the cleavage furrow, which help of myosin and actin. They contract and they pull these two apart. Now, one question which in my mind, most of you might be thinking that plant cells usually do not have centrosomes. So how do they help to form microtubules in order to make them move apart? Well, I did a little bit of research, and if you think there's any better answer, I'll be glad. So you can just write in the comment in the video. And it's that in a plant cell you basically have in a plant cell you have what we call the pre-prophase band. Pre-prophase band. And what it is it's actually an array or a network of microtubules surrounding the nucleus. So if you have this, so this is the pre-prophase band right here. It's just a big array of microtubules, and these actually during the mitosis expand like these, like if they are like, so these things which I'm drawing right here are these microtubules, and this is the nucleus. So I'm trying to draw it in a 3D, and I'm not successful in it. You can see that. So if you look at here, so now in this plant cell, these microtubules expand, or I don't know any better word, these microtubules like expand, these microtubules, this pre-prophase band expands, and this is these microtubules are eventually help the plant enduring mitosis. So they do not have centrioles, but they have this preprofase band of microtubules and network of microtubules which expand in this shape, in like an eye shape, and which helps the contents of the nucleus to flow in the opposite direction, such as the chromosome. So this is pretty much it, and hope you guys found it useful, and see you guys in the next video. Bye bye.